Hello class, today we are going to talk about the cell physiology. This is something that is both present and it takes place in various parts. It can be from the plant section and also it can be also in the animal sector. But specifically for our lesson today, we are going to talk about the plant physiology. From our previous classes, we have also dealt with the cell physiology, which majorly focuses about the study of the function of the cell structures. These cell structures are evident in plants and they include the cytoplasm, the cell membrane, and also the cytoplasm. But we shall talk about more as we continue with our lesson. The membrane structures and properties will be the first, and a membrane is a surface structure which encloses the cell and organelles. This is essential, and the membranes regulate the flow of materials into and out of the cell or organelles. Organelles, these are the organs present in a cell. The examples of membranes include the cell membrane, the nuclear membrane, and mitochondrial membrane, and the chloroplast membrane. The cell membrane, it has got 3 layers, 2 protein layers and phospholipid layer sandwiched in between the two. The properties of the cell membrane include semi-permeability. Semi-permeability is the ability of the cell membrane to allow movement of substances into and out of the cell. It has small pores, allowing for the passage of molecules of small size into and out of the cell. The cell wall, however, allows all materials to pass through it, hence referred to as being permeable. There is also sensitivity to changes in temperature and pH, whereby extreme temperature and pH affects the cell membrane since it has some protein layers. Such changes alter the structure of the cell membrane, affecting its normal functioning. Possession to electric charges. It has both the negative and the positive charges, helping the cell to detect changes in the environment. These charges also affect the manner in which the substance moves in and out of the cell. There, we are also tackling the parts of the physiological process as the introductory part. The physiological process, these are the ability of the cell to control the movement of substance in and out of the cell, and is achieved through the physiological process such as diffusion, osmosis, and active transport. This one also takes place in the plants, and for example, for the instance of diffusion, it involves the movement of molecules from a region of high concentration to a region of low concentration. This one we can also find it in our day-to-day activities, which can also include carrying out experiments in order to see the further results. Osmosis also involves the movement of particles from a region of low concentration to high concentration. This also is evident in various experiments, which we can also tackle in our day-to-day activities. Let us begin with diffusion. Diffusion is the process by which particles move from a region of high concentration to a region of low concentration. And from our practical activity is to demonstrate the diffusion using the potassium permanganate 7. This potassium permanganate 7 is added to a beaker containing water, and from at first it is in its particle nature, but after some time, for example 5 minutes, we find out that the colorless liquid turns into pink. This is due to the process by which the potassium permanganate will diffuse into the water, that is from its where it is highly concentrated to all parts of the beaker where it is low concentrated. And the difference in the concentration particles between the region of high concentration to the region of low concentration is known as the diffusion gradient. This diffusion plays a crucial role in living organisms, which begins by absorption of materials. Plants in their day-to-day, they need uptake of materials for their growth and to carry out their physiological processes. The mineral salts in the soil enter the root by diffusion, since their concentration in the soil is whereby they are high than in the root hair cells. And the digested food, which is the glucose and the amino acid, diffuse across the wall of the ileum into the blood for transport to the rest of the body. This is in living things, which can also include the human beings. We have the gaseous exchange in the plants and also evident in the animals. In both plants and animals, respiratory gases, which are the oxygen and CO2, they are exchanged through the simple diffusion depending on their concentration gradients. This is also evident whereby the plants taking the CO2 and give out the oxygen gas, which is consumed by the human beings, and this is dependent on each other. We also have the factors affecting diffusion. We have the diffusion gradients. The greater the diffusion gradient between the 2 points, the more the rate of diffusion also increases. We have the surface area to volume ratio, whereby the higher the ratio, the greater the rate of diffusion, and the lower the ratio, the lower the rate of diffusion. This also means that small organisms expose a large surface area to the surrounding compared to large organisms. Therefore, the small organisms depend more on diffusion as a means of transport of food, respiratory gases, and waste products. We have the thickness of the membrane and the tissues. The thicker the membrane, the lower the rate of diffusion, because the distance covered by the diffusing molecule is greater, and the thinner the membrane, the faster the rate of diffusion. We also have the size of the molecules. The small and light molecules diffuse faster than a large and heavy molecules. From our practical activity, we can be able, for example, you have a cube shaped, let's say a piece of let's say we are comparing that is a piece of chalk. When it's in its whole nature and added to a glass of water, it will not diffuse faster compared when it's in its crushed state. We also have the temperature, whereby increase in the temperature increase the energy content in molecules, causing them to move faster. The thickness of the membranes and the tissues. The thicker the membrane, the lower the rate of diffusion, because the distance covered by the diffusing molecule is greater, and the thinner the membrane, the faster the rate. Let's go back to osmosis. This is also a physiological process as the diffusion. It is more of the same, it's only that we have the difference in variation is minimum. Osmosis is the process whereby the solvent molecules, that is the water, they move from low concentrated solution, that is dilute, to a highly concentrated solution across a semi-permeable membrane. The highly concentrated solution is known as hypertonic solution. This can be the solution of a salt solution and sugar solution. Solution of the same concentration are said to be isotonic. This is a result of there is no difference in their highly concentration or low concentration. Osmosis is a special type of diffusion because it involves the movement of the solvent, the water molecules, from their region of high concentration to a region of low concentration across the semi-permeable membrane. The practical activity can be taking a yam. This yam can be taken and a hole can be made at the center. And at the center we can add either salt solution or the sugar solution, which is hypertonic, and we can place it in a beaker. That is its environment. The environment surrounding the yam will be the hypotonic environment. Therefore the water will move by osmosis from the beaker to the yam, and by the end of the day we shall find out that the solution, which can be the salt or the sugar solution, has formed in liquid form from its previous solid form. We have the osmotic pressure and osmotic potential related to osmosis. Let us begin by osmotic pressure. Osmotic pressure is the pressure which needs to be applied to a solution to prevent the inward flow of water across a semi-permeable membrane. This is the pressure that is needed to nullify osmosis. And osmotic pressure is measured using the osmotic potential. The osmotic potential also, this is the measure of the pressure of solution which will develop to withdraw the water molecules from pure water when separated by a semi-permeable membrane. We also have water relations in animals. The cell membrane of the animal cell is semi-permeable. This is different from those of plants. They are also semi-permeable, but in animals we lack the cell wall. And just like the dialysis, which is the whisking tubing, the cytoplasm contains dissolved sugars and salts in solution form. If an animal cell, for example a red blood cell, is placed in distilled water, which is hypotonic solution, water flows in by osmosis, and we have two processes that will take place, that is hemolysis and crenation. Beginning on hemolysis, the cell would swell up, eventually burst, and because the cell membrane is weak, The bursting of the red blood cell when placed therefore in hypotonic solution is called hemolysis. Crenation is similar, it's only just that the solution is hypertonic, and when the red blood cell is placed in hypertonic solution, it will draw out water and it will shrink finally. Body fluids that surround the cell must therefore have the same concentration as to that which is found inside the cell to avoid further crenation or hemolysis, which can cause dysfunctioning of the body. And we also have water relation in plants. When a plant cell is placed in hypotonic solution, it gains water by osmosis and descends outwards. But since it has the cell wall, it prevents it from further bursting. As the cell gains more water, its vacuole enlarges and exerts an outward pressure called the Tucker pressure. As more water is drawn in, the cell becomes firm and rigid and is said to be turgid. The cell wall in plant cell is rigid and prevents the cells from bursting, unlike the case in animal cell. The cell wall develops a resistant pressure that pushes towards the inside. This pressure is equal and opposite the Tucker pressure and is called the wall pressure. When a plant is placed in hypertonic solution, also physiological process which is plasmolysis and deplasmolysis take place. Water molecules move out of the cell into the solution by osmosis. The cell shrinks and becomes flaccid. If the cell continues to lose more water, the plasma membrane pulls away from the cell wall towards the center. The process through which the plant cell lose water, shrink and become flaccid is called plasmolysis and can be reversed by placing a flaccid cell in distilled water, and the process is called deplasmolysis. Wilting activity also takes place in plants, and also it's important for our discussion today. Wilting occurs when plants lose water through evaporation and transpiration. Cells lose turgidity, they shrink, and the plant droops. This is called wilting. And if water supply from the soil is inadequate, plants do not recover, hence permanent wilting. And this mostly takes place where there is inadequate water supply. The role of osmosis in organisms includes absorption of water from the soil, that is from the root hair cells of plants, which absorbs the water from the soil by osmosis to the rest of the plant's parts. We also have support. Support is essential in plants, and for example the cells of herbaceous plants, which are less woody, they absorb water, become turgid and support. This is more different compared to the woody plants. And opening and closing of the stomata is also essential. That is mostly come in relation to photosynthesis, and the stomata plays a crucial role. For example, during the day, the gut cells synthesize glucose. Therefore, they draw in water, become turgid, hence opening the stomach. During the night, they lose turgidity since there is no photosynthesis. As a result, they shrink, thus closing the stomach. We have feeding in insectivorous plants. These plants also can be compared to the case of the carnivores in animals. Their diet depends on the carnivores. So these plants are able to change their target pressure on the leaves, which close, trapping insects, which are digested to provide the plants with nitrogen in order to survive on their day-to-day basis. We have osmoregulation. This takes place in humans at the kidney. In the kidney tubules, water is reabsorbed back to the body by osmosis to prevent dehydration. We also have the factors affecting osmosis. Let us begin with concentration of solution and concentration gradient. The greater the concentration gradient between 2 points, the faster the rate of osmosis. Optimum temperature as long as it does not destroy the semi-permeability of the membrane. Active transport is a process that moves substance across the cell membrane against a concentration gradient, and this process requires energy to move the substance across cell membranes and involves substances such as the amino acids, the sugar, many ions, and which are taken in by the living organism through the active transport. The role of the active transport include reabsorption of the sugars and useful substance by the kidney, which can be used in the body processes. We have absorption of some mineral salts by the plant shoots, which we said earlier, from the root hair cells to the rest parts of the plants. We have absorption of digested food from the alimentary canal into the bloodstream, which plays a crucial role in the nutrition in the humans. We also have accumulation of substance in the body to offset osmotic imbalance in arid and saline environment. This can be evident in the mangroves. We have excretion of waste products from body cells to get rid of the harmful substance in order for our body to carry out its processes on its normal basis. Factors affecting active transport include the oxygen concentration. We have the changes in pH, which can be from its acidic, basic nature. We also have glucose concentration and enzyme inhibitors. And also, to take note on the same, any factor affecting energy production affects the rate of active transport. That is all we had for today. Bye bye, and see you next time.